What kind of sandwiches do you have?
Just a like panini crust regular sandwich.
It looks good.
That's fun. That's fun.
There we go.
Okay, good afternoon. One of the labs yesterday, we were talking about spring classes, people needing an extra class, and they said, oh, you ought to consider death and dying. They were like, what?
Oh, it's good.
Yeah, Jackie has taken death and dying the last time it came around. So if you had to describe like what it's about, apart from the official, what would you say that it's about?
Learning the different ways and cultures that that we think about death and how like there are different ways to do burials and ceremonies. Actually, very interesting. Ways you can cremate your body or like bury your body that are natural or not natural, like be a tree or like your ashes get mixed into the cement of the coral reef that you're trying to do. It's really cool. I recommend it.
Yeah. So this comes around every other spring. This coming spring is one of the semesters is offered. And for you all, it would be an elective unless you're doing the BA in biology, human bio track, and then we actually count it as part of a degree requirement. But almost all of you, in your career, you're going to be working around death. And maybe you haven't really thought about that part very much, but having an understanding of like traditions and rituals around death and how people process death, that's another part of this class, can be really useful for your career. It's not a high workload kind of class. And if you need a class to fill up your schedule, this would be a good option. So it's a 4-credit class, and the number on it is Religion 2800. This is with Dr. Alderman, who's our chaplain. And he is, I don't know, if you don't know him well, it's a healing to be in his presence, I would say. And like a really good person to get to know. And he has written recommendations for students that came out of this class, like as they apply to different programs and so on. Those of you going into physical therapy, some of those patients are going to be passing away, like as you treat them, or they will be experiencing in some ways the death of their abilities, which is somewhat related. So this is just a really good class. It meets in the spring on Tuesday, Thursday, 11:30 to 1. So if you need to pick up a class, or if you have some other placeholder class in your schedule and you think this would be preferable, I would highly recommend it. It's a really good course. So I wanted to give a little blurb for that. It's still got space. It's a lot of discussion type of course. And I think you all were supposed to do more field trips than you did last time. Yeah. But maybe this spring, field trips will be more possible. Okay, so the next thing I want to do, I need to give back your labs from yesterday. If you could not come to lab yesterday, you do not need to make it up. It'll be fine. You just be like missing that grade, like it didn't happen. But for those of you that could come, we're going to use these handouts to kind of relate the lecture topic today. These are not, yeah, they are an ABC. You can turn yours back down. Just keep it. If you get this back and you have something drastically wrong on it and you want to redo it, you can turn it back in and have it rephrased. That is up to you. If you missed lab yesterday and you really want to do this and have a good lab grade to put in with your average, you also can do that. Just let me know after class. I just said the whole the first time this happened. That drives my voice crazy. All of them get called the name of the whole I'll just turn it over. On the chart, what I did on that is I looked at your list of, is it resistant, intermediate, or susceptible? And if your answers are matching what I had, I don't go look at the numbers. If you have one of the resistant, intermediate, susceptible, and it didn't match what I have, then I go and look at your measurement. And if it's within 3 millimeters of my measurement, I'll count that okay. If you have some drastically different measurement number, that's when I circle and then I put minus whatever, because I think you're off a little bit. So you didn't have to be exactly, you had to be within a certain range. Like 3 millimeters is the range of accuracy there. So that's how the chart went. And then as I go through today on this lecture, certain questions, when I get to it, I will discuss it at the relevant time in the lecture. And any redos that you want to do, go ahead. But basically, I want this to be a grade improver if you want it to be a grade improver. So, but if you're okay with it, just you don't have to turn it back in also. So last time we had talked about how do we classify these antimicrobial drugs and name them in different ways, and what are their modes of action. And we had 5 different modes of action. So one of the modes of action is to block the synthesis of the cell wall. And that is the one that penicillin does. Penicillin is in that class. So on your worksheet on the number 2 question, explain why Proteus might be resistant to penicillin using your knowledge of its cell wall structure and penicillin's mode of action. So we know that penicillin is a cell wall inhibitor. And what it's specifically doing is it's blocking peptidoglycan action, let's say. So Proteus vulgaris is gram-negative, and it's got cell membrane. It's got thin peptidoglycan. And then it's got outer membrane. And when we put a drug into the environment of a bacterium, the drug is outside. So the drug is on the outside. So for penicillin, the target is the peptidoglycan. And in a gram-negative cell, it cannot go very well through that outer membrane, and it can't get to the target as well. So gram-negatives are not very susceptible to the original penicillin like we were using yesterday in class. But, so Proteus, and also Pseudomonas, which you tested yesterday, they would be in that situation, and penicillin is not harming them. But something like Staph aureus that's got thick peptidoglycan outside the cell membrane, and it's gram-positive. If the drug is in its environment, it can directly touch the peptidoglycan and mess up that cell wall a lot easier than a gram-negative cell wall. And we saw on the chart from yesterday that that was a big zone. Staph aureus had a very big zone with penicillin P10. Was it 10? Yeah, P10. So what a drug targets has to do with what cells it's useful for. And that kind of relates to this chart also. So here's penicillin G, the original, and that's what we were using yesterday. And it's narrow spectrum. It would be narrow spectrum because gram-positives would what it would work best on. But if you kind of read down through this list that increases by, okay, we're coming up to more modern drugs, more expensive drugs, more modified drugs. Notice at this point, ampicillin, it's a version of penicillin, has been modified to be broad spectrum and to work on gram-negatives. So what ampicillin has, so penicillin cannot get through the outer membrane, but ampicillin, which is more modern and more expensive, has a modification that lets it go through a membrane and then get to peptidoglycan and now start working on gram-negatives. So that was a lab modification that happened, I want to say like in the 70s. It's not a new drug, but it's not as old as the original. So when we take these drugs and modify them in the lab, you can give them chemical modifications, like through chemistry, to make them do new things and get to the target better. So that's kind of related to question 2 on there. So not every drug is going to be useful against every strain. Certain strains are resistant to certain drugs. And that's kind of our topic today, like how do strains get resistant and what can we do about it to help that problem? So if a strain or a certain kind of microbe doesn't even have the target of the drug, that drug is not going to help. It's not going to do anything. And we would call that situation natural resistance. Like Proteus vulgaris has natural resistance to penicillin G, let's say. Or something like SARS-CoV-2 has natural resistance to rifampin because it doesn't even have the target that rifampin targets. So not every drug is good for every infection. If a strain develops resistance, if a strain develops resistance to a drug that used to harm it, that's more of a concern to us. We don't need every drug to work on every strain, but we need a certain set of drugs to work on a certain set of strains. So if a drug used to be susceptible to, if a strain used to be susceptible or attacked by a drug, and then it changes so that it's no longer attacked by the drug, we call that the development of resistance. So that would mean that it stops, or the drug stops working on that strain when it used to work just fine. So this question about where does resistance originally come from is addressing if a strain that used to have a good drug against it, now that drug's not working, how does that happen? And here's a cartoon example of that. So we have a collection of bacteria in a broth, like we would in lab. And in this collection, let's say that they're all Proteus vulgaris. And we put those on an agar plate that's just a standard agar plate. And if we were to look at the whole population of those cells, the white ones are not drug resistant, so they would be killed by the drug. And the ones in red have something special about them that they would resist the drug. And normally, those ones in red that are resistant, that has come from a mutation. So that's coming from a mutation in the strain, or let's say a mutation or a change. In the strains, I'm going to be more broad because it could be a virus that doesn't have DNA. In the strain's genetic material, something has changed in its genes that affects either the target of the drug or some kind of protein involved with the drug's process, let's say. So something about how that drug interacts with that microbe has changed from a genetic mutation. That's where new resistance comes from. And that just kind of happens at a low frequency. It's not common, but that happens at a low frequency. It would be a rare event to get those, but they exist. So in a usual situation, those drug-resistant bacteria do not have any advantage over their neighbors unless what? When does it help you to be drug resistant? If you're allergic to something, helps to be drug resistant. Here's a silly example. So imagine that I am, like I'm actually an X-Man, something special like that, or superhero, and I'm actually resistant to X-rays. But I didn't know it because I've never been around an X-ray. So I cannot show my quality. That it doesn't help me to be resistant to X-rays until I'm around X-rays. And then I become like, oh, that's going to help me now. So being drug resistant does not give an advantage unless, if you've got these slides, circle this word, antibiotics added or drugs added. You can be resistant to a drug and it doesn't help you really unless suddenly you get in a drug-rich environment. So same population, if you put that onto an environment where you do have the drug and you look after an hour, so all the ones that are not drug resistant are going to die because the drug's killing them like it should. Those two that have that mutated new resistance, they're not going to be killed. And if the whole population is being exposed to the drug, guess whose descendants are going to take over the earth, the tube? It's the resistant ones. And this is just kind of a biological fact. Like if suddenly the temperature outside was 150 every day, like I would definitely not going to make it. Kids not going to make it. There might be like 3 people in Tennessee that could deal with that on a daily basis. And if they met each other and had like the hottest babies, you would end up with a population of people that can live at 150 degrees every day. Because that's what's advantageous. That's who's going to survive. So, resistance to drugs happens through mutation, and mutations are just going to happen at a low rate. But when you start to see really a lot of drug-resistant microbes is when you have a lot of drugs existing in the environment. Which leads to like a really depressing fact, which is this. Every time that we use antimicrobial drugs, or really any kind of killing strategy against microbes, we are generating what we would call selective pressure for the resistant organisms to predominate. And this is not just a medical concept, this is an ecology concept and through all types of life. So one of the funniest examples of this that I read recently is somebody that was doing a study on squirrels that live in cities. And they had tracked over time that when squirrels live in cities and they eat trash, because there's all this trash that they could eat instead of going and eating acorns or whatever they do, that the squirrels that are better at digging through trash or whatever it is, like that's a selective advantage. And that over time, squirrels as a whole in cities are getting better at eating trash. That's an interesting study. It's a similar concept. So if we use, as soon as we start to use an antimicrobial drug, we start to select for drug-resistant microbes. And in our history of types of penicillin, the older versions, something you could add on to this slide if you're keeping a list of like, this is old and this is new, and this is cheap and this is expensive. The older it is, the more drug resistance there is against that strain. So these days, it is not even very common anymore to prescribe the original penicillin G because so many microbes have become resistant to it. The ones down here, there are fewer microbes resistant to these new ones, but it's just kind of like, it follows that eventually microbes can mutate to avoid being killed by these drugs, which is a bummer. Okay. So that's where resistance comes from. It's going to spread more if we're using more drugs. So the ways that resistance spreads are the ways that any genes spread. So this would go underneath there. Erase this. Drug resistance spreads by the same ways that any genes spread. Methods of gene transfer. So the first and most obvious one is descendants of the original, let's say. So if you're a strain and you become resistant to the drug, you would assume that all of your descendants, offspring, the rest of the population that comes from you, they would also be resistant. So that's a way that resistance increases. That's maybe obvious, but worth listing down. And then the other ways that genes are transferred are the ones that we learned about back in Unit 2, which would be transformation, conjugation, and transduction. Those can spread any kind of genes one place to another, including drug resistance genes. So that is actually why we care about these medically, is because microbes use them to pass genes to each other. And the more useful the gene, the more advantage it gives, the more likely it is to spread between different strains. So you might think, well, like, okay, there are these resistance genes. What actually are they? There are 5 different categories of what resistance genes can actually be. And all of them are shown on this screen, but I know that these tables are hard to read. So the first type of resistance gene, the one that is the best studied and the oldest known, is an enzyme called penicillinase. That's what that says here, penicillinase. And it's a member of a class of genes that literally destroy the antibiotic drug. So class 1, we have enzymes. So the gene would make an enzyme. So we'll call this genes that encode, let's say. You got DNA or RNA that encodes production of these. So class 1, enzymes that destroy the drug structure. So, example, penicillin ACE. It's a penicillin cutting enzyme. And what it does is it cuts that bond right there. And that seems like boring from a chemistry sense, but it's the difference between this drug treating your infection or this drug not doing a thing for your infection because the bacteria is able to cut it up. That's pretty cool if you have that, if you're a bacterium. This is like the best chart in the whole book right here. Keep going back to it. So if you have an enzyme that cuts this bond, it's likely that it's going to cut that bond in all penicillin drugs because they all have this basic same drug structure. Come back to that in a minute. So that's class 1. Class 2. There's a gene that encodes some kind of change to a protein that controls drug entry. Like the drug is there, but it can't get in where the microbe is. So in this scenario, they're showing the drug is a little blue ball, and it has to bind onto a protein on the surface of the cell that then lets it come in. So if the microbe changes the shape of that receptor protein, the drug can be out there, but it's not getting into the cell. Penicillin does not work that way. It actually acts outside the cell. So that's not a good way to resist penicillin. But if the target is inside the cell and you don't let the drug in, fine. Like it'd be great if we could seal up all the buildings and not let the smell in. Think about that. There's actually a guy coming today to talk to the faculty about the smell. And like, I'm glad it smells. Like, see, it smells. It's a problem. So imagine if all the buildings were, sealed up, and it wouldn't matter how much it smells out there if it can't get in here. That's like this method of resistance. The third one is my favorite. I find it really funny because I'm a dork. This just strikes me as funny. So it's a pump that immediately pumps the drug back out of the cell. And this would be like you wanna go out on the lake in your boat and your boat has a leak. Right. Yeah, I'm gonna go anyway. I'm just gonna keep bailing it out the whole time I'm out there. So bail it out. You can keep your boat afloat as long as you keep on pumping the water out of it. It's the same concept. So yeah, okay, there's a drug. It comes into the cell and it's in there, but you have a gene that makes a pump that just kicks it right back out of the cell. I find that funny. This one is kind of rare. There's not that many drug pump proteins that can get the drug back out of the cell. But it's a cool mechanism. The fourth, I'm actually going to teach you the 4 ways. We're going to ignore this one. The fourth way is that the shape, there is a new and altered shape of the target protein that the drug can't bind to now. So the drug, say if the drug is supposed to get in the cell and get to a ribosome and bind to the ribosome and mess up its function. If the ribosome changes shape a little bit at that spot, the drug will no longer bind. That one's pretty common. that concept of, okay, the shapes are supposed to bind and now they don't, that's what everyone's concerned about with this new Omicron variant. Like, is the spike different enough shape that our antibodies aren't going to bind to it? And we don't know yet. And so like a week ago, I was very worried about that. So far, I'm less worried, but it's going to take about a month before we really know that. So I'm going to live the next 3 months like our little boys got their second shot on Sunday. It's like, okay, for 3 months, we're like, we've done what we can do. Everybody got all their vaccines, and I got my booster. So we're gonna move on, and then we'll just have to see, is our antibodies that you already have against COVID gonna bind to the shape of this new variant and destroy it, or are they not? So that's the number one COVID question of the week in the science world. So this are like what kind of genes would give you this drug resisting capability. So I need to say something about like, why is drug resistance bad for us? So the more strains that become resistant to drugs, the fewer options we have for treating people. So I want you to, if you got your chart, like refer back to your chart here. And I picked out 3 particular strains that we use in lab: Staph aureus, Proteus vulgaris, and Pseudomonas. And these are just standard strains. Like, these aren't special drug resistant strains. And question 3 on there said, which type of strain appears to be the most difficult to treat and why? And it's definitely Pseudomonas aeruginosa, because out of our list of 6, there were only 2 drugs on that list that could treat it. It resists the rest. And that's kind of true in the actual world of Pseudomonas treatment also. There are not that many drugs that treat it. So every drug that you lose to resistance means more costs and fewer treatment options for your patients. And there are some particular drug-resistant strains that I want you to have heard of. 2 particular categories that are particularly dangerous, and it's been a problem for years, and it's getting worse. Which are the acronym MRSA, M-R-S-A. You pronounce this MRSA. And then a category called VRE. So MRSA and VRE. What are some of these? Say that louder. Yeah, it's a staph infection. So the SA is Staphylococcus aureus. The R is resistant. And the M stands for methicillin. So what MRSA stands for is methicillin resistant Staph aureus. And if you have a MRSA infection, it is a staph infection, but you are infected with a version of staph that resists, excuse me, that resists methicillin. And it's not just methicillin. It's the whole penicillin category. So it was first observed to be resistant to methicillin, but it resists the whole category. So resist methicillin and all of the other penicillin-based drugs. And there are certain MRSA strains that have also acquired more resistance against more drugs. So patients that have a MRSA infection have very few drug options available to them. And the drugs that they do have available, they may have to take it by IV. Or it might cause them really bad side effects, or it might cost $1,000 a month and stuff like that. So it's a real problem. There's an equivalent category called VRE, which stands for vancomycin resistant Enterobacteriaceae. And remember that this is a category of gram-negative gut bacteria. And it would include E. coli. It basically includes all of our gram-negatives from lab except Pseudomonas would be considered in this group. And this group is resistant to vancomycin and related drugs, which reduces your treatment options. So antibiotic resistance is a real problem. We need to try to not encourage more of it to happen. And that's what this slide is set up for. So what can we do about drug resistance? And this will be the last one, last slide for this particular day. Some of these strategies are going to relate to like what individual people can do, and some of it's going to be like governmental level stuff. So, number one, I need a meme of that guy. Like, if you could blah 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 blah, that'd be great. It's from an old movie called Office Space. It's really funny. Okay, so number one, prescribe the actual correct drug for the actual infecting microbe. That is the best way to not overuse antimicrobial drugs and encourage drug resistance. What's the drawback with that? This doesn't happen all the time. Why not?
Hard to get the correct dosage.
It can be hard to get the correct dosage. It's expensive. It is. How is it expensive? What's the expensive part of that?
Hard to narrow it down to a single line.
Yeah, like how do you know what is infecting that patient? Like, I think maybe a few of you identified your unknown in like 2 days or something. If you happen to get like start immediately. I didn't say you, I said if you, you immediately start to work and you gram stain it and you have to grow it overnight and stuff like that. If you think about a patient coming in your office and they've got a problem and you've got 20 minutes with them, maybe. Like, how are you going to figure out what's wrong with them? Our office visit system is not really set up that way to immediately identify the strain. So for this one, identifying the strain takes time and money. And if you're going to take the time and the money to do that, it means that you're not going to give the patient a drug until you figure out what's wrong with them, and that tends to make them upset. And then they're also kind of, got to wait for relief to get the right drug. You ever been to, maybe you don't notice this yet, but like been to a doctor's office, and it used to be that they would have they got a prescription pad. And like, it's already filled out with like, okay, this is for amoxicillin for X mg per mil, taken orally for your child that has an ear infection. And I drank gallons of that stuff as a child. I had so many ear infections. And when my boys were very little, they had those sitting in the doctor's office, like ready, like you rip one off, you sign it, like here you go, got an ear infection. And they're not doing that now because of this. It's better to know what the strain is. But here's an alternate case. You have a super bad UTI, you go to urgent care, you pee in the cup, they say, yes, you have a UTI. Here is a prescription, because you're suffering, and it's probably one of 3 things that this drug will treat. So this happened to me 2 years ago. And then I was so happy. The next day they called back and they said, don't take that anymore. It's not going to work. Go to the pharmacy and get this instead. So what they did was they took my pee cup. There you go. Looks like a muffin or something. And then they cultured on a plate like we just did yesterday with the top 6 drugs. And this is the one they gave me. And here's the bacteria growing up against it. Like, well, that's not going to work for her. Call her back. Tell her to get a prescription for this. So that's kind of a middle path. Like, okay, try to give somebody something that you think will work, but then also test the bacteria, see what's going to work. And if you were wrong in the first place, go update their prescription. That's a good middle path. Otherwise, I would have been taking this, not clearing the urinary tract infection, and then just encouraging resistance to that useless drug of the other microbes that are in my body. So that's a really important one. And that will come up in your career most likely if you're a nurse. Like, you're probably gonna have to deal with somebody, why can't I have maybe they don't talk like that. Why aren't you prescribing me this? That's what my last doctor gave me. Well, maybe that's not what your problem is today. Okay, second one, which will also relate to your nursing career a lot. So you get your prescription, and maybe it says you're supposed to take these 3 times a day for 8 days or something like that. And you start taking it, it's actually working, you feel good. You get to day 3 and you're like, yeah, I think I'm good. It's better to take the whole prescription and not leave like lingering halfway resistant microbes that are maybe just kind of barely hanging on. Go ahead and take the whole prescription. And don't save that drug for the next time. This is something that definitely older people do. And I bet you've got people like this in your family. I do. They'll drag out a pill bottle from 5 years ago. Like, oh, I've got some of this. Here you go. Should not be doing that. That's going to be hard to change for the older generation. But it's really important to encourage people to take their medicine the way that they're supposed to. That also goes for pain pills. Like, there's not pain pills to get stolen if people take them as prescribed. Or if they're not going to take them, they're supposed to destroy them. So encourage your patients to take all their prescription as indicated. Okay, number 3. And this one is not going to relate to you in your direct career, but it's important. Some of the biggest use of antimicrobial drugs in this country is not for people, it's for animals on a huge scale, like in factory farms. So Where this happens, like having the drug in the water, it's not in test tubes. It's in sewer systems, and it's in lakes, and it's in puddles, and it's in whatever water where microbes live. So environmental antibiotics also contribute to this, not just when it's in our body. So reducing agricultural use would be helpful also. Here's another one. We're not separate from other countries in, like, we don't have border control for antibiotic resistance or COVID. I mean, countries think that they do. Did you see today that, like, oh, we shut the border, everybody's shutting their borders for this Omicron variant? And like, it already was there. It already was in Europe a week before they shut the border. You just don't know it. How do you find it? So it matters a lot what all countries do, not just this country. Let's write this in a more positive way. Okay. So some countries, including, well, I don't even name the countries. Here in the U.S., it's kind of hard for just your average person to go get some antibiotics because they want some. You have to be prescribed them. And maybe it's not too hard to get prescribed them if you're really determined about it. But some countries, you can go to the store and you can buy antibiotics for really cheap because you think that you need them. That's kind of like, whoa, that's weird. But other countries, certain antibiotics, you can do that. And then, I don't know, like, is it people's right to take antibiotics that they don't need? That's an interesting question. It does contribute to drug resistance. And then I'm going to put a fifth one. These are not all the ways, but these are some top ways. And the fifth one is a governmental and an industry question. And I actually think this one is going to get better now. It's been very difficult to address this, but there needs to be more funding for developing new drugs. So for so many years, starting in the 40s with the OG original penicillin G, there was a lot of money put into, and research and effort putting into, let's find more antibiotic drugs so that we can really help infectious disease. And most of our major drugs that we have today started out being researched in the 50s and the 60s. And then we had so many, it was working so well. Yes, let's move on to things like develop Viagra and cholesterol drugs and stuff that's real profitable that people take all the time. So, and not just for a acute situation. So developing these drugs takes years to do and a lot of money. And private industry, you know, if your drug company, your major driving force is your shareholder profits, you're going to put your investment into drugs that make you a ton of profit, like Viagra, like Jardiance, which is people who are pre-diabetic. There's so many of them. Lipitor, stuff that's like, oh yeah, you need to take that every day the rest of your life. That's going to make you a lot of money. Whereas if you invest $180 million in a drug that could treat, tuberculosis and get rid of it in 3 weeks, and people are going to take it for 3 weeks, you're not going to make as much money off of that. So I can understand why private industry does not want to go into antibiotic development, because you take them for a few weeks and then you're done. And then we don't want to overprescribe them because of resistance. So it's not a great way to making money, but that's a place where governments can step in. And I think the COVID pandemic, if there's one thing that's not controversial about it, it was the concept that like, oh, we really could use some better drugs for this. That I think is an agreed upon area. And I think that government investment in drug design is going to increase all over the world. It's probably going to be for viruses first, but some massive event like this changes people's outlook. Like, oh yes, we need to invest in drug development now, so that in 10 years we'll have more drugs. Mm-hmm.
How do you think that like all these like organizations raise money for like cancer? For, like you still get 500 years.
Cancer is so difficult because like this is not going to be a satisfying answer, but you're trying to kill a part of your own body that has basically got all of the characteristics of your own body. But I do think in the next 10, 15 years that antibody-based cancer therapy, like immunotherapy, that's going to pay off a lot. It's not going to be pills. It's going to be IV of antibodies that they like custom design for your cancer. And they're probably going to be generated from your own cells. And like when you all start to get old enough to get cancer, that's going to be great. Oh yeah, it's expensive. It's super expensive. It's super expensive. And it requires genetic engineering. And you'll have to go to a clinic to do it, but it's like it's going to feel like chemotherapy when people go and get chemo, which is chemicals through IV. This is going to be antibodies. Yeah, cancer is a tough one because it's all your cells, and they might have 3 differences between their neighbor cells, and you don't want to kill the neighbors, but you want to kill the cancer. Yeah. I don't know. You guys, I'm not that much older than you. I should stop being negative and be like, by the time you, but you're coming into like a wonderful time for disease treatments, I feel. And I hope what you learn in here helps you understand them and how they're going to work. So, yeah.
I think the slides, because it's
Yeah, so Friday's lecture, it's got very few slides, but there's a math technique that we're doing, which will be on the final, but it's pretty easy. It's like fraction dividing stuff. So you'll need to bring, your phone, which is probably your calculator. But Friday in this class is pretty important topic, and it's few slides, but you do need to bring them in like your math brain. I'll teach you how to do this ratio dividing, but it's about how do you pick out a good drug and what dose.
And then are we gonna go over like what's better on final?
I did on Monday, so get those from
Oh, I
somebody.
should record it, but I have those.
Okay, good. Yeah. Okay, yeah, if you want to turn these back in,
Yeah.
please do
Except
that.
for maybe 1 or 2 slides, but I can.